All right, so yesterday we discussed the incomplete dominance pattern. So this is where there are two alleles, and the two alleles, when they work together, they form a new phenotype, a unique phenotype. In this case, the pink phenotype, and uh, it's because the two alleles, when they are in combination, there's some sort of interaction between them, or something, some sort of way, some sort of method or mechanism by which these two alleles, these two different versions of a gene, can function together to make some sort of unique appearance. And if we have only one allele, if we have a homologous case, we end up with something like a red flower. If we have a homologous for the other allele, we have a white flower. But when they work together, we get some sort of unique phenotype. In this case, an intermediate phenotype. So this is the pink flower in the snapdragons. Okay. If you find that the relationship is that there are two different alleles, but when they combine together, they form a new trait that has some features of both alleles, then this is known as codominance. Both alleles are working at full strength, and so we end up with some sort of combination of those two traits. So here we have a white allele that makes white fur. Here we have a red allele that makes red fur, and then when they are together, we get red and white fur. And so both alleles are active in these examples, um, in the heterozygous example. So that's a little different from incomplete. Incomplete, something you get a new trait or a new phenotype that's somewhere in between. In codominance, you have parts of the animal that have both traits, or parts of the cell that have both traits. It can really depend. And so we started discussing the AB blood type as an example of codominance. Here you have an A allele and a B allele, and when they work together, you create an AB blood type, which has both traits of the A and the B allele. And so the immune system would identify these AB blood as both A and B type. It doesn't identify it as a new type, it identifies it as both A and B. All right, so the ABO blood system comes from a system of more than two alleles. It comes from a system of three alleles. And so this is the multiple alleles pattern. So this is a pattern where you have more than the normal three genotypes. So I was just trying to work out the math here. So if you have the consequence of multiple alleles is that you have more than, I should actually just say, more than three genotypes. That saves me all of this little nonsense here. And so, for example, if you have three alleles, then you have 9 different combinations. How many of those are unique is what I was trying to figure out. So let's do a little bit of combinatorics here. You've got a 3 by 3, right? So A, B, C, A, B, C. We've got 1, 2, 3. Those will all be unique. And then 4, 5, and 6. I believe it's 6 different. It'll be 6 unique genotypes. I'm just going to get rid of this. So for example, if you had an A and a B and a C allele, then I'm just going to show you what you might have. You would have possibly an AA or an AB or an AC. You could have BB, BC, and you could have CC. These ones would all be mirrors of the other ones, so just be AB again. AB, better write them out for full explanation here. BC. And so we end up with 1, 2, 3, 4, 5, 6 different combinations. So that's a possibility of 6 genotypes if you had only 3 alleles. Now, fortunately, or maybe not fortunately, because actually this would probably be more simple to work with. Um, so I guess unfortunately, you also have to consider if the different alleles have relationships to each other that are similar to the other patterns. So, for example, in the ABO blood group, there are A, B, and O alleles, uh, typically labeled with this system here, where we label the A allele with a capital I and a little a and a capital A at the top, a capital I for the B allele, but with a capital B, and then a lowercase I is the O allele. And the reason we use this special notation is because the A and B alleles follow a codominance pattern. So A and B are codominant to each other, but A and B are dominant, follow a standard dominance recessive pattern to the I allele or to the O allele. This is our O type. So when you cross someone with A-type blood and someone with O-type blood, the A-type allele can overwrite or mask the O-type blood. But if you cross an A and a B-type person, then the two alleles can work together to form a codominance pair. And so in this way, instead of getting the possible 6 different types of alleles, 6 different types of phenotypes, we only end up with 4 unique phenotypes. We have the homozygous genotype of AA, which of course gives us the A type because it only has A genes. Or if you have a genotype that is heterozygous for A, but heterozygous with respect to the O allele, you also end up with A type blood. Homozygous for B gives you B type blood. Heterozygous for B with respect to an O type allele will also give us B type blood. AB blood is the result of the codominance heterozygote, and O type is only from the homozygous for O type. So that's definitely one of the most interesting gene pairs that we can have, and one of the most interesting systems to study as far as sort of answering questions and things related to this stuff. So I will post up some questions in a second about that. Now, as far as printing goes, is it okay if do you want also the assignments printed as well? Or are you okay with doing those on by hand on a piece of paper or something? Any comments on that? Okay, let me know about that. You're muted if you're answering, so. All right, so let's look at the cross. Here we have a parent that has IAI, so that means that this parent has which type of blood? A type blood. We remember that the A type is type. You can see how they've tried to indicate this with the way that they've labeled things. So notice that they've labeled things with the capital I, lowercase I, to show that the A type blood is dominant over the little I. I forget what the dominant I is supposed to stand for. It does mean something, but I'm forgetting it right now. I think usually around this time in the class I get Google out and I search it, but we'll see. If anybody wants to look up what is the capital I in A-type blood, that would be helpful maybe. Maybe not helpful. So we're going to bring down the allele from the top and the allele from the left. Bring those into that square. We're going to bring down the allele from I'm going to do the left one first because it's the dominant one, and the allele from the top, which is the recessive one. Allele from the top, which is the dominant one, and the allele from the left, which is recessive, and the two alleles from the left and top. So crossing two heterozygote pairs still gives us 4 genotypes, which is exactly what we would get normally, except now all 4 genotypes have are unique, and so we end up with a new ratio, right? This is a new ratio. We have 1 codominant pair to 1 dominant A to 1 dominant B, all the way to 1, oh, ran away on me, 1 homozygous recessive. Or another way of writing this is to say that we have 1 AB blood to 1 A blood to 1 B type to 1 O type. And so this is our phenotypes. And this is the only time that we have ever seen 4 unique phenotypes. Every other example that we've had had only up to 3 phenotypes. So we've gone from 2 phenotypes in pure Mendelian to 3 phenotypes in the intermediate or the incomplete and the codominants, up to 4 phenotypes in this case of the multiple alleles. So there you go. So that's a sort of a fun one, and you get a lot of different. There's a lot of different interesting questions we can ask about this one. So let's look at this example. If a woman has AB blood and a man has type A blood, what possible blood types could their children have? Well, there's 2 factors to consider here. The father could be homozygous for A, or they could be heterozygous for A and carrying the I gene or the O type allele. So we need to consider 2 different crosses in this case. The mother, who is AB blood, they are the easiest to look at because their blood type is defined by their phenotype, or their genotype is defined by the phenotype. So I'm going to put the mother on top, and I'm going to make my little square here, and I'm going to make another one over here, and make another square, and then I'm going to put the father over here as the homozygous one, and then I'm going to put my father here with the recessive trait. Cross them for that first one, cross the second one, cross the third box here. I notice that I'm dealing with a homozygote, so the number of options is going to drop quite a bit. It's going to be halved. And then in the other case, I have a heterozygote, so I am going to get some interesting options here. And there's the I, and there's the I. So, there's an interesting result here. If you were to ever study this family and you found that they had one child who had the B-type blood, it would actually inform you that the father must be heterozygous for that I type, for that I, because this is the only time that we see that B-type blood appearing. So here we got a 1 AB to 1 A type, or a 1-to-1 ratio, because we can see that there's 1 and 1. And in the second one, we see that we have 2 A-type blood with 1 AB blood and 1 B-type. So that's a really, that's not a unique ratio. We have seen the 1-to-1 before, but it is unique in this case because of the sort of format of those ratios. There is a chat if you in this function as well in Meet if you want. It's if you're using your computer, it's up on the top, a little sort of chat icon. I think on the phone, it's also on the bottom maybe. So if you want to drop in questions there, or if you want to stop me at any point and ask me questions, that's cool too. Okay. Anybody have any questions about ASAP, and we can have maybe a class of going through some of those problems.
I think I zoned out for a part of it. What was the I representing?
Yeah, so the I represents the O allele. So O is recessive to the other two alleles. So to represent it as being recessive, it's in the lowercase, in a lowercase I, and then the A and B have both been sort of have been attached to this I allele to represent that they are dominant over the other allele. Does that make any sense?
Yeah.
So there's three different alleles. IA blood type meaning. I forget. It means something. That big I. It means like. Yeah, I can't quite remember. It's on the edge of my tongue. That's why it's bothering me so much. Is like I remember looking it up. I look it up every year. I feel like this image is actually really useful. I don't think I have it in here, but I'm gonna add a page and see if I can find it real fast. Oh, there we go. This is when everything is connected. Life works well. So this is sort of the image that I would use in my Bio 30 class a lot. Is to think about the blood groups like this. Here you have. We'll throw on our little bits of information here. So we have the four different types of blood. This group here is going to be the IA, or IAI. And in this case, what's happening is that as long as you have one allele for the A type protein, you will be able to produce these A type antigens on the outside of the cell. If you have a case where you have the other type, so let's just make them green so they stand up a little more, where you're carrying those B alleles. In either case, in either the heterozygous case there or in the dominant case, then you will be able to produce these B-type antigens. If you are a carrier for both of the alleles, then your cell will produce both of those alleles on its outside. And if you only carry the recessive allele, then you get nothing. Nothing, no proteins form or no carbohydrates form on the outside of the O-type blood. So the O-type blood is empty. And that's one of the reasons why you can have a heterozygous case, because in the heterozygous case, you've got one allele that produces protein and one allele that produces nothing. And so when they are combined together, the cell still produces protein because it has one functioning allele. The other allele, the little I, doesn't do anything. It just sits there, and it's not particularly useful. And this is one of the ways that O-type blood became so popular in the world, is that I think overall O-type blood is like 46% or something of the global of the world population. I'm looking at for that data. Blood type. Global blood distribution type. And so O-type is the most common. 63% of the world is O-type. So it's actually really, really much higher than I was saying. I'm always surprised at how high it is. And one of the reasons that it's so high is that O-type blood is uh, O-type blood is the result of one of 6 different mutations. So I will post this link here. There's a little interesting link I've looked at before, and it has the percentage of the population that carries the A or B allele, and then the population that has O-type. And in South America, for example, 90 to 100% of the population is O-type. So that's pretty wild. You know, uh, that really shows you kind of what we're talking about here. Here's the distribution of O-type blood. And I believe in our evolution section, I mentioned how O-type blood has a certain prevalence because the group benefits from O-type blood. So O-type blood does not have antigens on the outside, so it's much easier to use for mixing blood. If there's any mixing of blood, there's no reaction to that blood. So it has a high it can spread through a population pretty quickly because it's a fairly efficient it's an efficient mutation for keeping a group alive. The antigens don't really impact the individual, but they do benefit the group. So yeah, and it's also can be the result of different mutations that all result in the same phenotype. So it doesn't actually have to be, it's like the population over here in Africa that have O-type blood aren't carrying the same O-type blood that the population over here are. It could have been independently, what do you call that, parallel evolution. Um, of those two groups because it can be the result of multiple different mutations. So even we might even think about these little pockets of I believe there are actually pockets of mutation in different areas, and how the population spread from those areas outwards could be the result of different mutations at those sites. Don't know. I don't have an anthropological knowledge of it. I just have some basic fundamentals there, but certainly something that could be researched and is being researched all the time. Okay, I got one more, and then we're gonna break for today because the next ones are the tricky ones and the most modern types of inheritance. So the next one is called the lethal gene, and this is a case where certain gene combinations do not have a survival rate, or they don't survive. So certain gene combinations can be deadly to the point where those genotypes cannot reach sexual maturity to removing them from the gene pool. So this changes the ratios that you might. This changes the ratio because the result is going to be that when you look at your offspring, you'll be missing some, and you might not even realize that you're missing them because the gene combinations are so deadly. So this was a case where there was these mutant agouti mice. So these were mice that were carrying a mutant gene. They were lab mice, and the mutant gene caused them to turn yellow. And so they were just an interesting case of these yellow mice that they had mutated in the lab, and they thought, oh, well, this is really cool. We have mutant yellow mice, and we can make them non-yellow when we mate them. But when they were crossing them, they kept crossing them, and they said, it's a little strange. We're never getting close to that. We would expect that sort of 3/4 to 1/4 ratio, or maybe a 1 to 2 to 1 ratio, one of the other Mendelian ratios. But instead, they kept discovering a ratio of 2/3 to 1/3. And so this 2/3 to 1/3 ratio was something they never seen before, and they couldn't figure out what was going on, because one of the gene combinations they realized was turning out to be a deadly combination at the fetus level. So when the baby mice were being born, there were some of the what do they call them, kits, offspring. Some of the offspring were either didn't even make it past like the zygote level, or they were being stillborn, and so there's just a sort of an abnormally high level of stillborns in these certain crosses of these yellow mice, and they didn't put it together that it took a little research to put it together that these mice were the carriers for a certain gene combination. And so when they looked at the phenotype ratios, it only made sense if they included a genotype that was not present in the living offspring. So from the gene pool, moving them from the gene pool. Maybe gene pool is not the right word there, but okay. So the offspring phenotype ratio is altered by the missing deadly So, for example, so in this case, it was the homozygous dominant. If mice were born with the homozygous dominant gene, they would die. Usually, this death was beyond, like, even earlier than this stage. I believe that they don't even make it past the zygote stage, or maybe a blastula stage or something. So the result was that you would have only the two genotypes that could survive. And so looking at our cross here, we have a heterozygote and a heterozygote, which gives us normally a 1 to 2 to 1 ratio. But if you have to eliminate this one, they died off, now you have a ratio that is 2 to 1. So you have a 2 to 1 ratio of surviving offspring. And that's why that's the lethal gene combination, and why that has a unique pattern. Now, there are a couple of interesting human examples that we can pull. Some of them don't even really apply anymore because science has gotten so good, or medical science has gotten so good. So, for example, for a long time, you'll still find a lot of references to sickle cell anemia as being a lethal gene. Being born with sickle cell anemia would often mean that you might not make it through childhood. Today, with modern medicine, we're definitely able to have a much higher survival rate for sickle cell anemia, and so it's not really considered a lethal gene. It's an unfortunate gene combination, but it's not considered lethal anymore because of modern medicine. Similarly, Huntington's disease is a dominant lethal allele, but it usually only becomes deadly in later life. So an individual may have children before the disease manifests. So it's a fringe case of lethal genes, because it doesn't actually affect the phenotype ratio. It just affects but it does affect the individual, and their chance of living a full life is reduced if they are born with that Huntington's. I believe it only takes effect in the late 30s. So it can be a very sad case of an early death. So if one parent is heterozygous for Huntington's and the other is not a carrier, what is percentage of the children may be afflicted by the disease? So let's take that. Let's do this example here. So here we have Huntington's, and we have one that is heterozygous for Huntington's, and then in another case where we have a parent who is not a carrier. Now this is an interesting one because if we did use the dominant gene up here, but it was, in this case, it was only one combination. If we say that Huntington's disease is dominant lethal, then I think we're suggesting that if you are, this person here is going to develop Huntington's in their lifetime. Just saw a typing error here. So we can see that that someone who has Huntington's, who chooses to have children, they, even if they're only a carrier, they have 50% of their children of offspring will likely develop the disease. So yeah, this is part of the idea of genetic testing before you choose to have a child. This is one of the things that that people will test for, because often people are thinking about having kids in their 20s, uh, or earlier sometimes, and sometimes a little later. But having a genetic test beforehand for some of these diseases, or thinking about family history, are some of the things that people consider before deciding to have children, if they decide. Sometimes it's not a decision, right? All right. Well, anyways, with that, those are the next two inheritance types, and we'll move from here on to the really weird ones, which are the intergene inheritance types. And those ones will take a little bit of extra work to discuss, but I am looking forward to discussing those with you tomorrow. And so I'm going to end this.